So good afternoon, everyone. It is such a great pleasure to be here today. I really enjoyed interesting presentations by the previous speakers and also the thought-provoking discussions. I'm especially touched by the kind words about FDA by the previous speaker, Dr. Grimes. Today, I will talk about the role of model-informed drug development for COVID-19 from a regulatory perspective. It is somewhat different from the previous talks, but I think it will be another important step for drug repurposing for the current and future pandemics. I will use one specific example we recently published to discuss the role of model-based analysis in drug development for COVID-19. I'm also happy that Dr. Grimes talked about hydroxychloroquine because this will be the focus of my talk. It is about how to connect hydroxychloroquine in vitro antiviral activity to in vivo drug concentration in order to predict the potential antiviral effect in human. I put a conclusion up front here. That is, under a safe oral dosing regimen, it is unlikely that hydroxychloroquine can achieve high enough concentration, in vivo concentration, to show antiviral effect in human. I will explain how we reached this conclusion in this talk. This is a timeline of the FDA announcements related to hydroxychloroquine. In March 2020, FDA issued the emergency use authorization to permit the emergency use of hydroxychloroquine for treatment of COVID-19 in certain hospitalized patients. Then in April, FDA cautioned against the use outside of the hospital setting or clinical trial due to risk of heart problem. In May, our paper was published. On June 3rd, FDA highlighted our publication through a CDER Spotlight article. Then on June 15th, FDA revoked the EUA for both hydroxychloroquine and chloroquine. In the June 15th letter to revoke the emergency use authorization for chloroquine and hydroxychloroquine, FDA listed 4 reasons as the basis for this decision. The first one, the suggested dosing regimen for chloroquine and hydroxychloroquine, as detailed in the fact sheets, are unlikely to produce an antiviral effect, is based on our publication. Next, I will go over some background information about hydroxychloroquine. Hydroxychloroquine has been approved to treat malaria, RA, and lupus. The mechanism of action for malaria indication is proposed to be related to the drug's weak base feature. Similar to chloroquine, hydroxychloroquine may exert its effect by concentrating in the acid vesicles of the parasite and by inhibiting polymerization of heme. It also inhibits certain enzymes by its interaction with DNA. The mechanism of action for RA and lupus is unknown based on the current product label. The approved hydroxychloroquine doses for the 3 indications are all different. The highest daily dose is for the treatment of uncomplicated malaria. It is 800 milligram plus 400 milligram on day 1, followed by 400 milligram on day 2 and day 3. So the total dose on day 1 is 1200 milligram. The daily dose for the other 2 indications are lower, but the treatment duration is longer. Under the chronic use condition for RA and lupus, it was highlighted that the incidence of retinopathy will be higher if the maintenance dose is exceeded. This is a list of safety warnings on the package insert for hydroxychloroquine. I already mentioned the eye toxicity, but the cardiac toxicity may be more well-known because of all the news reports. Even though this talk is about hydroxychloroquine, the science is relevant for both chloroquine and hydroxychloroquine. Both drugs are weak base and have extensive tissue distribution. The hydroxychloroquine product label mentioned the terminal half-life ranged from 40 to 50 days. The long half-life can be attributed to the extensive tissue uptake. Similarly, chloroquine label mentioned that in animals, tissue concentration could be 200 to 700 times the plasma concentration. This is a very important feature that will be extensively discussed in the next few slides. During the early phase of this pandemic, two papers reported, sorry, my slides jumped. Okay. During the early phase of this pandemic, two papers reported the in vitro antiviral activity of both hydroxychloroquine and chloroquine. The results on the left is from New et al., and the results from the right is from Yao et al. The top panel shows the raw concentration response data, and the bottom panel shows the estimated EC50 under different conditions. The EC50 is the concentration of a drug that gives half of the maximum effect. It is commonly used as a measure for the drug's potency. Among all the EC50 estimates under the different conditions, the lowest value was 0.72 micromolar. We will use this lowest EC50 for all discussions because this represents the most promising scenario for hydroxychloroquine's in vitro antiviral activity. Yao et al. Also built a PBPK model based on literature data for both chloroquine and hydroxychloroquine, and used this model to predict both plasma and lung tissue concentrations. The plot on the left is a plasma concentration, and the plot on the right is a lung concentration, which is about 400-fold higher than the plasma concentration. The pink line is for chloroquine under the 500 milligram BID dosing regimen, and the other lines are for the different dosing regimens for hydroxychloroquine. I only listed regimen B and F here because they are the closest to the EUA regimen of 800 milligram on day 1 and 400 milligram once daily for 4 to 7 more days. The authors compared the predicted free lung trough concentration with the estimated EC50 of 0.72 micromolar, and this table summarized the ratio under the different dosing regimens. As we can see, the ratios is well above 1, especially for hydroxychloroquine, suggesting the in vivo tissue concentration of hydroxychloroquine should be significantly higher than the EC50 value, suggesting a robust antiviral effect for all these dosing regimens. The similar conclusions was reached by many other papers. I listed a few here. All these papers linked a much higher tissue concentration with a reported in vitro EC50. For example, one of the paper concluded that it is plausible that a single dose of 400 milligram or even 200 milligram can provide adequate lung tissue concentration to inhibit the virus. Here are 3 more papers. The first one find that the simulated unbound lung concentration were above the EC50 for all evaluated dosing regimens, including a subtherapeutic 10 milligram dose. Now, I would like to use a plot to explain the common problem with all these papers. The top panel here shows the in vivo experiment condition, and the bottom panel shows a corresponding in vivo condition. The EC50 reported in all this paper is based on the added drug concentration in the cell culture media or the extracellular space. Due to the weak base feature of chloroquine and hydroxychloroquine, the drug will become ionized once it enters the acid vesicle in the host cell, such as lysosome. Once ionized, the drug gets stuck inside the intracellular acid compartment, and this is the reason for the high tissue concentration. It was reported that chloroquine can be enriched up to 1,000-fold relatively to the extracellular concentration in vitro cell culture condition. Similarly, the much higher in vivo tissue concentration relative to plasma concentration is due to the same reason. More importantly, we know the tissue concentration is not homogeneous. the lung interstitial fluid concentration or the tissue's extracellular concentration should be much lower than the lysosome concentration, and the free tissue extracellular concentration should be similar to the free plasma concentration. Remember, the EC50 was all estimated on the extracellular concentration in cell culture media. No one used the much higher intracellular concentration to calculate the in vitro EC50. Therefore, the correct in vivo concentration that should be compared to the in vitro EC50 is a free lung extracellular concentration or the free plasma concentration, not the much higher tissue concentration that is mainly driven by the intracellular concentration. If we use the correct in vivo concentration to recalculate this ratio, all the numbers were approximately 400-fold lower. If we use the EC50 estimates from the Liu paper, the ratio will be even lower, suggesting the in vivo relevant concentration is too low to have antiviral effect. If we put the in vivo concentration on the original concentration response curve, the red line on the x-axis will correspond to almost no viral inhibition on the x-axis according to the observed data. However, all the papers discussed earlier linked the much higher concentration range indicated by the blue line with the in vitro experimental data, which we think is an apple to orange comparison. It turned out that there are many tricky issues for this drug. Some publications directly compared the blood or serum concentration with the in vitro EC50 value. In fact, hydroxychloroquine is also accumulated in the different blood cells, meaning in thrombocytes and granulocytes. As a result, the whole blood concentration is by average 7-fold of plasma total concentration. Even the serum concentration is about 2-fold of plasma concentration because during the blood clotting process to generate the serum, some of the drug in the different blood cells diffuse back to the serum. This was extensively studied by some scientists back in the 1980s. These papers helped us a lot to interpret findings from various trials. For example, this paper reported the individual level concentrations. Even if we take the maximum concentration points among all these individuals taking the 200 milligram of hydroxychloroquine 3 times a day, all of the correct concentration, after correct conversion, the free plasma concentration is only about 0.47 micromolar, lower than the lowest reported EC50 of 0.72 micromolar. This paper is a well-known paper that contributed to the enthusiasm of the hydroxychloroquine. The efficacy data was heavily criticized because of the long randomized design and exclusion of some patient data. We took a look at the PK data in this paper. If we convert the serum data to the free plasma concentration data, again, the median level is only half of the lowest EC50 value of 0.72 micromole. To make things even more complicated, the centrifuge force is very critical for the proper process of the blood sample. Again, scientists figured this minimum centrifuge force back in the 1980s. 1,000 G was recommended for chloroquine, and an even higher centrifuge force of 1,200 G was applied to get reliable plasma concentration of hydroxychloroquine. One study applied only 250 G to process the blood sample and significantly overestimate the plasma concentration of hydroxychloroquine. In fact, their reported plasma concentration in this paper was as high as the whole blood concentration. The overestimated plasma concentration was used to develop a population PK model for hydroxychloroquine plasma concentration. This PK model was used by one of the earlier papers to predict the plasma concentration and the corresponding lung tissue concentration. As a result, this problematic PPK model combined with the inappropriate lung tissue concentration led to the conclusion that even 10 milligrams of hydroxychloroquine could generate high enough in vivo concentration for the antiviral effect. In fact, multiple papers relied on this misleading PK model to predict hydroxychloroquine plasma concentration. So it is really critical to use reliable data to check the validity of the reported model before directly using it for prediction. Despite all the problems we just discussed so far, some researchers got this right. Here's a paper by a group of scientists from Duke University. They were trying to figure out a safe and efficacious and effective dose for hydroxychloroquine in the pediatric population. They realized the exact same problem as we did and concluded that the simulated unbound hydroxychloroquine concentration in interstitial fluid mirrored those in unbound plasma concentration and were notably lower than the in vitro concentrations needed to mediate antiviral effect. Therefore, the in vivo concentration under safe hydroxychloroquine dosing is too low to have antiviral effect. Remember, this conclusion is already based on the lowest EC50 value of 0.72 micromolar. When more and more groups were repeating the in vitro experiment, some groups realized that the cell lines used may not be appropriate. The most commonly used cell line is Viral or Viral E6 cell line, which is a monkey kidney cell line. When this group compared EC50 values estimated from Viral cell line and other cell lines such as CALO3 cell line, a human lung cancer cell line, they find that EC50 for the CALO3 cell line for both chloroquine and hydroxychloroquine are much higher as highlighted in this slide. For hydroxychloroquine, it increased by 30-fold, and for chloroquine, it increased by 5-fold. The hypothesis is related to the different expression level of TMPRSS2 in different cell lines. Two recent papers published on Nature further confirmed these earlier studies. The second paper here included extensive animal studies and concluded that hydroxychloroquine did not show a significant effect on viral load in any of the analyzed tissues. So in summary, for hydroxychloroquine, an in vitro antiviral EC50 value reported in the literature where extracellular drug concentration present in cell culture and should be compared with in vivo free drug concentration in the plasma instead of the much higher tissue concentration that is driven by the intracellular concentration. Under the assumption that in vivo cellular accumulation is similar to that from the in vivo studies, the calculated free lung concentration that would result from proposed dosing regimen are well below the in vitro EC50 values, making the antiviral effect against SARS-CoV-2 unlikely with a safe oral dosing regimen. Since we only considered viral inhibition activity, hydroxychloroquine may have additional relevant pharmacological response, for example, anti-inflammatory or immunomodulatory effect. However, well-designed clinical trials failed to find any effect in a studied population. I would like to end my presentation with a reflection on this case study. Since the beginning of the COVID-19 pandemic, significant effort by scientists around the world to identify drugs with antiviral activity against SARS-CoV-2 has led to drug development at an unprecedented speed. And the necessary step to this process is translating in vitro antiviral activity to appropriate clinical dosing regimen. This step is complex and multifactorial, but essential to finding the right product administered at the right doses to the right patients at the right time for successful treatment of COVID-19. Finally, I would like to thank all my colleagues. This was a great teamwork with a very short time frame. Without their dedication, this work would not be possible. And thank you for your attention.
Great, fantastic. Thank you so much, Ki, for this great presentation. Again, it was wonderful to see how you brought our attention the challenge and importance of modeling the right concentrations and selecting the right dosage for the right patients. There are some questions, so let me start with the first one. So David Brown is asking the following. He says, I think you did not mention zinc. One opinion is that the antiviral activity of hydroxychloroquine alone is not the relevant factor, and instead hydroxychloroquine acts in the way that it helps transport zinc into cells, and then zinc changes the pH of levels of the cell and reduces ability of the virus to replicate. Some of the papers you quoted do not focus on this. Can you comment or elaborate any aspect of this question that David had?
I acknowledge there's still a lot of information that is unknown about hydroxychloroquine and other drugs that is currently under development. So I don't have any specific knowledge regarding zinc per se, but my general philosophy is all this hypothesis will need to be tested, maybe first in the right in vitro cell culture models, and maybe in the animal models, and in the end, we will need to link the right concentration from the in vitro experiment with the in vivo PK concentrations in patients, and then come up with the right dosing regimen, and then test finally, like the most likely the randomized clinical trials will give us the most definitive answer.
Okay, great. There is a follow-up comment from David. He says the failed clinical trials used hydroxychloroquine alone. They did not co-dose zinc. Any comment on?
Yes. Yes, again, like I understand, acknowledge there are many different like hypotheses and theories, like many different beliefs. But in the end, like before we like approve any potential therapy for patients, we really need to have convincing data from the clinical trials to really understand how well the drug works. So that's all I can say at this point.
Great, thank you. I have another question, which is more general question that potentially many computational scientists have in the audience or ask during over the day, related to availability of the information on dosage or appropriate concentrations that failed at different type of experiments. So many of the data sets that we are pulling for computational models come from public sources of biorepositories that do not have dosage or concentration information. So how can you comment a bit whether there are opportunities for people to access more detailed information on concentration, dosage for various drugs in the context of COVID, or more broadly for other diseases and drugs at your center?
Yeah, that's a great question. I think to understand the pharmacokinetic information is really it's a very critical step when we try to predict the in vivo antiviral activity for all these agents based on in vitro data. And it is true, like when we rely on literature data for the PK information, there are a lot of challenges as we discussed today, like even for chloroquine and hydroxychloroquine, when you look at different literature, there are some papers using like a whole blood concentration, some using the plasma concentration, some using the serum concentration. You can see there's a lot of actually difference in those concentrations, and there's not to mention there will be a lot of actually variability related to for example, like intersubject variability, etc. So in general, in the ideal scenario, if in a drug repurposing condition, like if we could use the PK information that is used for the drug clinical trials for drug approval, this kind of PK information is probably of the highest quality because the companies will be required to conduct like bioanalytical validations, and FDA will be reviewing those validation data and to make sure the PK data are reliable before we're using it. And some of those data are in the public domain, for example, at the FDA website, such as Drugs and FDA, where our review for those approvals will be made available to the public. So if those kind of information can be used, that would be great. We also like we also know there are many like PK information in the public literature, and sometimes it's hard to know like because in for FDA, when we review the PK information, we always request validation reports. But for the literature articles, they do not always have the validation data set. So in the end, when we look at those data, we will take into consideration all information, but we will be like, be careful about not to overemphasize the validity of one article over another.
Great, thank you for clarifying. Another question that I have related to getting the data from published scientific articles. There certainly is. Are there computational strategies that you use? That do you get the data through manual curation or search, or are there computational methods and automatic extraction of these data sets? And a second question is, how do you then do you have statistical methods to account for various confounders that might be present in a particular data set, or numbers that you get from a particular paper?
Okay, so at FDA, because of our role is mostly related to drug development phase and regulation, so we usually do not do a lot of work in the drug discovery space, but like quantitative like a data science and modeling approach has been used very often in the drug development space, and also like our regulatory review stage as well. And for example, as a clinical pharmacologist, we very often build mechanistic like models for both pharmacokinetics and dynamics, and try to integrate information together to predict what will be the drug response for a certain drug at certain dosing regimen. And this is actually very routinely used for like all of our reviews for each like NDA and BLA application, like pharmacometrics review will be integral part of that. And in terms of understanding the confounders, and that is a very important factor as well. In fact, as a clinical pharmacologist, one of our key mission is to understand the various intrinsic and extrinsic factors that will impact the drug exposure as well as drug response. For example, different age, gender, ethnicity, organ dysfunction, concomitant medication, or other like comorbidities, etc. So whenever when we analyze data, we always try to address those confounding factors as well. I hope this answers the question.
Yes, it does. Thank you so much. There is another question from the audience, and that is, does FDA has a data repository on the PK data for approved drugs?
Yes, we do. Yes, for all new drug applications and BLA, which is for biologic license applications, almost all the drugs will have like clinical studies with pharmacokinetic information, and all those information will be submitted to FDA for review. So we do have the data repository here.
And the follow-up question is if the data repository is publicly available, meaning that if participants at today's talk could go to the website and get the data readily available, or is there a particular process that they need to go through?
So for clinical trial data, like FDA, like in the FDA database, those are proprietary information, so we have to protect the confidentiality. So those data are not available in the public domain. However, in the FDA review, very often we will have the summary level data available in our review, which are available in the public domain, but we don't have patient level data available for the public.
And so interested people can access the data through FDA website and your center at the FDA.
Yes, I think the website is probably called Drugs and FDA, where we will have like a data repository for the drug package inserts, the drugs approval history, the drug approval letters, and also the multidisciplinary review that is used to support the drug approval are available there. And actually, for some of the other, because at the website, most of the information that is available are for new molecular entities, and sometimes for supplemental applications, it may not be there, but it may be possible for people to request those information about FDA review through something called the Freedom of Information Act.
Okay, great. Thank you so much for these clarifications and pointers to some of the data resources that you were mentioning during the talk. And thank you for this presentation.